Hi, welcome to another entry of the Learn Adventurously webinar series. Today we're going to talk about how to, visualize biological data using ggplot2. So this is a topic that is near and dear to me because ggplot2 has helped me dramatically, and I think it's the most widely used visualization software out there, maybe aside from default Excel. So let's just start off with what is ggplot2. And what we're going to do in this webinar is we're going to cover the very basics of ggplot. We're going to teach you how to actually plot your data, give you some best practices. And then at the end, we're going to leave some room for questions and answers. So please feel free to talk in chat, ask questions in chat, and we'll address all of them at the end of this webinar. We'll leave plenty of space. And I am just now realizing that I have a filter up. So green colors may be a little weird. We're just going to roll with it. So what is ggplot2? So ggplot2 is an R package that enables users to make custom visualizations. So we will be working in R today as well. I have a wonderful blank RStudio set up. We won't cover in depth how to actually use RStudio, how to download all that. That is a completely different concept. We are just going to focus on ggplot2 today. Now, if you have any material, this is where I want to say, I'm going to say this right up at the start. I have a full course that includes how to get started in R, but also has a whole section on ggplot2. We actually have that pre-loaded up. So again, I sell many courses. Yeah, I don't know why I have a green filter on, but yeah, the greens are very weird, right? So I can actually fix that in 1 second. Filters, here we go. Very weird. I don't know why there was a chroma key on that. Haha, there we go. So yeah, I do have a course, Fundamentals of R for Biologists. I promise I won't sell for too long, but we do have a full section on ggplot2. So there are, about 6-7 lessons, quizzes, video lectures that just teach you, how to use ggplot2. And if you are not willing or not able to pay, we also have a free ebook that you can download, which covers the material, just doesn't have all the video lectures and everything. But I do want to say that because you're showing up to this amazing webinar, if you just use the code webinar, it's active right now. You can get a 20% off of all of the courses. But I'm not going to worry. I'm not going to sell you anymore. We're going to dive right into it. And we are going to be using the iris dataset today. So this is a default dataset in R. And what we can do is we can load it very, very easily. If we just type in data, iris, we're going to run that. Over in our environment, we now have this data set iris. If you've looked up tutorials, you have probably seen this data set. It includes 3 species of flowers and their sepal length, sepal width, and petal length. So we'll be using this data to actually visualize everything, right? And whenever I make any type of ggplot, I really do follow 5 simple steps. And so what we're going to do is I'm going to very quickly go through these steps, and then we're going to go back and go through each of these with a little bit more detail. So step number 1 is to specify the data. And we do this with this ggplot data equals function. So in this case, right, if we're back in R, if we go into the source, if we want to make our first ggplot, the first thing we really need to do is make sure we have ggplot loaded in. So it's ggplot2. It is also loaded inside of the tidyverse package. If you just type in library tidyverse, it'll load ggplot. Make sure we run that. And then now we can actually start making our plots. We now have all of the code needed to plot our ggplot. So we're going to type in ggplot data equals, and we're going to use the name of our data. Again, over our environment, the name of our data is iris. So we will just type in iris. Now, an interesting thing happens if we plot this data. I just plotted it. It's over in my plot window, and you get this just blank square. This is the first layer, right? The problem is we have this data set. R doesn't know what to actually do with it. Doesn't know which variables we want to plot, doesn't know how we want to plot it, right? And so that's where we lead into step number 2, which is using AES to map variables. AES is short for aesthetic mapping. It connects our data to the visual elements of our plot. So what we are going to do is we are going to, inside of that ggplot function, okay, we're going to type in AES, add parentheses. And here, we're usually just going to specify what data goes on the x-axis and what data goes on the y-axis. Just like, kind of a review of basic plotting principles, you're going to want to put the independent variable on the x-axis and the dependent variable on the y-axis, right? So in this case, we're just going to put x is equal to, we're going to go with we're going to go with petal width. I'm just going to type in petal.width, making sure I have any capitalizations, and then making sure it's also spelled correctly. And then on the y-axis, we're just going to put petal.length. So this is the first thing. And again, we put this inside of aesthetics, okay, inside of that AES function. And all this does is it says, hey, we're going to put the petal width data on the x-axis, put the petal length data on the y-axis. And if we plot this data, you can actually see, again, in our plot window, we now have a little bit more than just that gray box. We can see that petal width is on the x-axis, petal length is on the y-axis, but we still don't have any data that is plotted. And that is because we need to go to, step number 3, which is adding layers. So ggplot works through layers. We add on information sequentially over one another. So again, we're going through these steps very quickly. We'll go back and make sure we actually cover them in a little bit more depth. But let's add just geom point, okay? So to add a layer, we go back to R, we add the plus symbol. So this just says, hey, we're going to add another layer. On the next line, doesn't have to be on the next line, but I prefer it on the next line. That's the standard. I'm going to type in geom point. And all this is going to do is it's going to say, hey, R, we have this data on the x-axis, we have this data on the y-axis. I want you to make a scatter plot with that data. And if we run it, go to our plot window, we can see, hey, look at that. It has plotted our data. Now, if you're making a very, very quick plot, this might be all that you need. We can very quickly see that, hey, petal width, petal length, it's all there. But most of the time, we're going to want to do a little bit extra, right? So that's where step 4 comes in. This is where we actually start to customize our design. Now, I'm going to spend a lot more time actually talking about how to customize the design, but you can do this on the layer itself. Let's say we want to change the size of our points, make them a little bigger. We're going to do size equals 5. I'm going to put this into the plot window first. And then if I run that code, you can see that now it plots the same data, just with bigger sized points. And you can change color, you can change the shape of the variables. If you're working with like line graphs, for example, you can change how that line is plotted. So again, these are very simple steps. All we did was we got ggplot in there, we specified what is our data, we added a layer, and now we're customizing it. And so once you finally customize it precisely how you want it, that's when you just figure out step 5, which is exporting the design. Now I'm going to spend a little bit more time on exporting design in these next few steps because there's multiple ways of doing this, and I want to make sure that we cover it properly. But those are really the 5 steps. Like this is what we do for every single time I make a ggplot. Specify the data, use AES to map variables, add layers, customize the design, export. And now is when we're actually going to go a little bit more in depth as to how to make this happen. So with step 1, that's specifying data, unfortunately there are so many different ways to get your data set up for ggplot, and that could be a whole discussion on itself. So whenever you're getting that data in there, just make sure it is good data. Make sure it is free of errors. Make sure that you're not having typos, for example. But I really want to focus in on AES specifically, because the aesthetic mappings of ggplot is where I find most of my students have the most trouble. And I think it's because it's very easy to make mistakes. So remember, AES stands for aesthetic mappings. It maps our data to our visuals, right? So just like here, where we put inside of AES, we said that the petal width and the petal length is going on the x-axis and the y-axis respectively. We can use AES in numerous ways, right? So one way that I really enjoy using AES is to actually have our plot be designed visually in a way that uses our data. What do I mean by that? So say we have our plot here, right? Well, in this data set, we have 3 different species of flower. How can we tell that from this data? This is really difficult to tell. So what we can use is we can actually use AES to better visualize this data. So inside of this geom point layer, I'm going to add AES again. Again, put it in parentheses. This is a whole different argument, so we're going to separate AES in size by that comma. And inside of AES, I'm going to say I want the color of those points that we added, I want them to correspond to the species. So again, we are mapping our data to our visuals. In this specific case, we are mapping the data contained in the species column to the colors of our plot. And go to plot and I run this code, we can now see that the data is colored according to our species, right? We have red points for the Setosa species, green points for versicolor, and blue for virginica. So this is a very, very common usage of AES, and it is a relatively straightforward procedure. But, unfortunately, there are numerous mistakes that occur around AES. And I want to say that one of the most common mistakes I see is forgetting that your data needs to be inside of AES. Okay, so in this case, like let's say we wanted to set the size of the points according to sepal length, right? So we're back in our code, we go to size, we're like, okay, we don't want it to be that standard set 5. We want it to be the bigger the sepal length, the bigger the points. In this case, let's actually use petal width since that's the data we're working with. Let's say we want to set it to petal.width. Make sure we actually use the correct capitalizations and all that. And if I run this code, well, what we get down in the console is an error. And we get this error because R doesn't know where this information is. It needs to be in aesthetic mappings to say that we are grabbing this information from our data set and we are using it on ggplot. Right now, it is outside of AES. And if we set it up, so let's now include it inside of AES, remove that little parentheses, and then add another one out here. So now color and size are going to plot according to our data. So if we go to the plot, I'm going to run that code. We can now see that, one, the color is according to species, but also larger petal widths have larger data points. And that is because we used our aesthetic mappings. So again, make sure you're always putting your data, if you want to use your data, put it inside of aesthetics, put it inside of AES. And there is a inverse problem, right? There's the opposite. Specifying color sizes or shapes that are not in your data inside of AES. This is another really, really common problem, and it's an easy mistake to make. I make this mistake pretty often. So every time I say common mistakes, these are my common mistakes, and I'm just assuming they're happening to other people. Let's say that we don't want to color our data according to species, right? Say we want to just make it the color blue. So we go in here, we type out blue, look at that, we expect our points to all be that wonderful blue color right there. We run this data, and our data is red. What happens here? So I used blue specifically. It actually doesn't matter what color I specify. So if I'm just going to change it off screen to green. If I change it to green, you can see that in the legend, the color says green, but it's not green. It's red. What's going on here is that R is going to look at that aesthetic mapping. It's going to look at that color argument, and it's going to say, okay, I need to find green. Well, green is not in the data set. So I'm going to make a variable. We're going to call all of the entries green, and we're going to use the default color. In this case, the default color is red. If we use 2 colors, it'd be red and blue, right? So this is a very common mistake, and it's just because green is not in value in our data set. It's just not inside of that iris data set, right? If we look at that environment, we don't have a column called green. So just make sure that if you're trying to specify a specific color, a specific size, anything like that, anything that is not your data, that it is just outside of AES. So I'm going to remove it from here. We're going to put it here, remove that extra comma. And now if I plot it, you will see that the color will change to green. Okay, so these are by far the most common mistakes I will see surrounding AES and ggplot at large. So with that just here's a great general rule. If you're trying to reference your data or your visualization, put it inside of AES. And if you're adding information not from your data, it goes outside of AES, okay? So this is a really general rule, and I find that this helps me a lot with identifying common mistakes and where things need to be. So we're going to move on from AES, right? And we're going to talk a little bit more about layers, okay? So ggplot uses layers, and in fact, they go on top of one another. So here, if we were going down this, we would have ggplot is the first layer. That's where it makes that gray rectangle, that default with no data. Then the next layer would be geom_point, so that's a scatter plot. Then the next layer would be a line plot, right? And they go on top of one another, okay? And there are so many layers we can use with ggplot, okay? The geom_bar layer will create a histogram of one variable. The box plot layer is used by using geom_boxplot. Scatter plots, geom_point, line of best fit, geom_smooth, right? So there are many, many layers we can do. This is what I was just referencing earlier, right? ggplot first creates the base plot window. Then we can add on a geom_point to plot the data as a scatter plot. Then on top of that, maybe we want a line of best fit, right? So we use geom_smooth, and this plots the line of best fit. But make sure you're understanding the order of your layer, because each layer is added atop of the last one. So take this example here in the left-hand side, we have ggplot, we do the geom_point, we do geom_smooth, and you can see, I made these points really big. You can see that the line of best fit that was plotted with geom_smooth is over the points on top of them, because it comes later in that workflow. If we switch the order, if we put geom_smooth first instead of geom_point, now the geom_points are on top of that line of best fit. So this is not a bug, this is actually a feature. Oftentimes we are layering information and we want to have some information on top of the other. So just make sure you're keeping this in mind whenever you are working with. You're working from, as it goes from top to bottom, it's sort of the inverse. Whatever's on top of your code is first, and then whatever is lower down in the ggplot pipeline is going to be on. So this is a really common layer, and I wish we could talk more about the different types of layers. There are loads of them. I believe the default ggplot has around 20 or 30 different layers, and there are numerous packages for adding on additional layers. I use one, I think it's called ggdendro for phylogenies, right? That adds a lot of information. So I want to talk a little bit more about the design. Okay, we're going to move on from the layers, and we're going to talk about the design. So there are many, many ways to actually design your plots, right? We already talked about a few. We talked about size, we talked about color. There are other design elements we can use. Some of the common ones are shape, which changes the shape of the points. So if we go back to our plot that we made here, let's say I'm going to make the shape. With the shapes, they are in colors. I will be sending out a handout after this webinar. It will be available that shows you all of the default shapes you can do. So in this case, let's just choose lucky number 13. And if we plot this data, you can now see that all these points are very, very hard to see. Let's make this color, let me make the color black, because it'll be a little bit easier to see. But yeah, see now you can see that all of these data points are circles with an X through them. If I change the shape to, I'm just gonna choose number 2, random. Now you see there's triangles. So whenever you are working with ggplot, whenever you are working with these different design elements, just know that there is pretty much always a way to customize it precisely how you need. Just look through the documentation. ggplot has a wide array of different ways to change data. And if there's anything specific, feel free to ask it as a question. We'll cover it during the Q&A portion. But most of the time, I am just working with, size, shape, color, and fill. Fill and color get confused a lot. Color just changes the color of points and lines, where fill will actually color in the different areas. So if you're making like a box plot, for example, fill would actually be the colors of those boxes, while color would be the color of the lines outside the boxes. You can kind of think of it as one-dimensional versus two-dimensional data. That's kind of what works for me. But remember, we can absolutely use data for our design. Use your aesthetics to actually visualize your data properly. Again, this is one where we're using the species to color our data, and this makes a much clearer visualization. So just making sure that there are many ways you can do. Now, we're going to talk about exporting. Exporting is seemingly easy at first, but there are very specific issues you will run into. So for very quick exports, I always say use the GUI. This should be number 2. I'm just now realizing. But what I mean by is, say that this is your perfect plot, right? This is the plot that you want to send to your advisor and have them send you an email with all these accolades saying this is the greatest plot I've ever seen. If you just want to do this really quickly, I always recommend just going to export, save as image. Okay, you can also copy to clipboard if that's easier. Sometimes I do that if I'm communicating in WhatsApp, and that way I can just paste it into the chat. But just save it as an image. This will open up a smaller. This will just open up a little window where you can choose the file name, choose what the width and the height, what type of format it'll be in, and then all you need to do is just save this, okay? So if I save this rplot, it's going to go to my working directory folder, and we have that, it'll display in photos right here. So again, that's a very, very easy way to export it. However, this is not the appropriate way to export if you are using this for your publications, okay? Whenever you are trying to visualize data for a publication, what we need to do is use ggsave, okay? And ggsave can be a little bit daunting for folks, okay? So what we're going to do is we're going to type out ggsave here. Okay. By default, ggsave will save the last plot generated in the plot window. Okay, always going to be the last one. So if you want to do it just like I have it here, where we plot the data first and then we use ggsave, that's totally fine. And all you'll need to do is say, we're going to call this file ggsave export.png. This will export our document as a PNG. So I'm going to run this code. It will export this image. And now inside of our working directory, I'm just going to go over to files. We have this right here, ggsave export. Smaller so we can see it. But you might notice an issue, okay? So look at this. We have the GUI. This image on the left-hand side is the one where we just clicked export image. And now the one on the right-hand side is done with ggsave. Well, the size of the points is different. The text size is different. It does appear to be in much higher quality, right? If we zoom in at these data points, you can see that the one used with ggsave is at a much higher resolution than the one on the left-hand side. So ggsave gets complex because your screen, the physical screen of your computer, may have a different resolution, right? I'm working on a big screen, but I have another screen over here that is much smaller. My laptop is at a different screen resolution, right? It has slightly different sizes. If I resize the window within R, that can change the resolution. So this is a very common complaint, a very common issue when trying to make our publication quality exports, okay? And in fact, in the webinar, I have a really great example. So this visualization really shows the issues well. The preview window, the ggSave export, there are different resolutions. So you don't want to rely on that preview window. And what I mean by preview window is inside of R, whenever we actually plot the data, it goes down to this by default bottom right window. Don't rely on this for your publication quality figures, okay? Within ggSave, you can specify the width and the height, right? So let's say I want to set the width of this plot to 10 and the height to 7, okay? And then we're also going to set a resolution. By default, it is 300 with ggSave. That is usually fine for most publications. If you're using a very large figure, you're going to need a higher resolution. So we're just going to specify this DPI as 500. Let me actually zoom in on that. Apologies. So you can see here that we set the width to 10, the height to 7, and the DPI to 500. By default, these units are in inches. You can change them to pixels, to millimeters, but by default, this is going to create a 10 by 7 image with a resolution of 500. So if I run this again, it just did the last plot that we made in our plotting window. And I am going to open this up. We saved it as ggSave export. Now we can see that our plot window has a much better resolution, okay? This is because we just made the image larger, okay? It is still very high quality. We can zoom in and not have our figures all pixelated. But this is something where you just need to play around with the resolutions. I really don't think, in my experience, I have not found a appropriate methodology where you can get this right the first time. I always have to just mess with it, right? And just play around until it looks good. So just set the width, set the length. You may have to change the point size. Usually I get it to something like this where it looks relatively good, and then I will make the text size larger. I'll maybe make the points a little bit bigger, right? So this is just something to keep in mind, is that ggSave can be a little bit unpredictable. However, it is almost a requirement to make high quality publication figures, okay? And again, after this webinar, there'll be a webinar recap up at learnadv.com, and there you can actually have a little bit more information about how to set these resolutions properly, how to play around with everything. Okay, so from this point of the webinar, right, we went through, we talked about the 5 steps, we talked about resolution. What I want to leave you with, because we've been having about 30 minutes now, I'm going to leave you with 5 best practices for working with ggplot, and then we're going to open it up to Q&A. And these are best practices that I find work well for really anything beyond ggplot, just visualizing data in general. So 1, use the correct visualization for your data and your story. There is a huge body of literature about what type of visualization to use. You'll find some surprising elements, such as pie charts are hated by the visualization community. And I agree, don't use pie charts, ever. They're just not needed. Use a bar graph. But this is something that is really intentional. I mean, don't just plot your data willy-nilly. Really think about what you want your visualization to convey and make sure you're using the correct plots, right? If you're looking at, if you're trying to compare 2 different sites, right, or maybe 10 different sites for the number of species in every single site, you probably don't need a line graph, right? You're not looking at change over time. You're looking at changes between sites. So in that case, a bar graph may be perfectly fine, possibly a box plot, depending on your data. Number 2, keep it simple. Everyone has seen a figure that has a lot of text, a lot of information, lots of stuff going on, and because there's so much going on, it is utterly incomprehensible. So I always say keep it as simple as possible. If you want to specify anything about the plot, put it in the caption, keep it in the body text, make a little note on the side. I always say simple is better. Ensure legibility. This is a personal pet peeve of mine. I review undergraduate theses, and very, very often I will see that their figures are illegible. The text is way too small. Everything should be readable and very, very legible. I kind of always say that you can't assume the publication that you're putting this figure in will actually have it at that huge size that you want. And you may be working with people that have their, they may be looking at your visualization on their phone. They may not have the best data, so it displays the image as a little bit more pixelated. So just try to make sure the text is large enough, make sure the points are actually visible. And that kind of goes back to keeping it simple too. The less elements you have, the less text you have, the bigger you can make all the other text. Don't use the preview window to fine tune. This goes back to what we were talking about with ggsave, right? Where whenever we exported the information, sometimes those exports do not look good, right? It skipped ahead. That's not what I meant to do. But yeah, so this is what I'm saying. So this is where someone might have thought this, they made the plot on the left, they were looking at the preview window. This actually is not that legible. I would make the text bigger, maybe make it a little bit simpler. But when they exported it, it looks like this. And so in this case, just don't use that preview window to fine tune your data. So things like making sure your text size is proper, making sure the colors are good, making sure placement of all your elements is proper. Don't use that preview window because you're going to have results like this, and you're basically going to have to fine tune 2 or 3 more times. So just usually what I do is I just export it and then edit it. I open it up in Photos and then edit it, export again, check it again. There are some fancier methods where it can show you it as a developer window. But I find it's usually more trouble than it's worth, in my opinion. So I just export it, open it up, make the changes, re-export. It's code, it's just ggSave, so you just got to run one line of code, and then you can fine-tune it. It really doesn't take that much effort. And then finally, that last best practice, we already kind of showed it, use themes. Themes are something I wish I had the time to talk about here. I do talk about themes in my textbook as well as in my online course, very briefly, but themes are incredible time savers when it comes to your code. And luckily, ggplot already includes a few themes. So here we go. So a theme, as you can see from this tooltip, allows you to edit every single element of your plot to your heart's content. If you want to remove the legend, you can do that with a theme. If you want your plot background to be a different color, you can do that with your theme. So I almost always start with theme minimal. So if we go back to our plot window, we have this plot here. I'm just going to rerun it, but only with that theme minimal layer added. And what you can see is that it has removed some of the elements. It has changed a few things. It just makes your graph a little better. If I want to change it to a different theme, I'm just going to choose, let's do theme void. See what that looks like. There we go. So now it has removed all the axes, everything else, and it's kept a very simple visualization. There are third-party, I guess everything's third-party. R is open source. There are other packages that include a ton of themes. There's one that includes themes that are standard in journalism, like BBC has their own theme, right? So use your themes because they make your life a lot easier and they do a lot of the heavy lifting for you. You can absolutely make your own themes. That does take a little bit of time, but it's totally doable. It's something that you're probably gonna have to do if you need to be publication. We will cover making your own themes in ggplot at another webinar, but unfortunately I'm already a little bit over time for what I said I was going to do. So let me see here. There we go. So thank you. So as a reminder, you can use the code webinar to get 20% off the courses. There's no time limit on that. I'll make sure if you RSVP, you'll get an email that mentions this. Next week, we will be doing another webinar. This one, we're going to talk about phylogenetic signal. Really, how do we understand species similarity through their common descent? It'll be here again next week, same time. And if you want to view anything, okay, the webinar recap will be posted on the Learn Adventurously website, as well as any resources we produce for this specific webinar. I do have a full R Markdown that I am working on finishing up that covers all of the information from this webinar and more. This will be up for free. You can can download this, it'll walk you through everything. But again, I have a free ebook that already covers a lot of this information in a lot more detail up on the website. So with that, I want to leave it for any questions, any comments, anything at all. This time is for you. I do see a question from Isabella that says, can I talk about manipulating data sets within R to make it easier to plot, creating groups within the data set to selective plot data, or changing all time data from 24-hour format to 12-hour format? I can, not in this session, just because there's so many different ways of doing it. Creating groups within the data is something you can do with the tidyverse package. There's actually a group by function. However, this is maybe a little bonus ggplot tip, something that I had to ultimately remove from the data because it was, or from this presentation because it was just a little bit too much information, was the concept of facet wrapping. I love facet wrapping. So let's say in R, let's say we have that plot, right? We have our wonderful, wonderful little plot right down here. Well, we know there's 3 species in here. Maybe we don't want to color them differently. We just want to have 3 separate plots for each of the species. What you can do in facet wrapping is you can use the little tilde, it's a little squiggly line, and then set out your, let me see. You can set out the name of the variable. So here, we're just going to do tilde species. And what it does, this is my favorite thing in R, is that it'll actually split it up into different individual plots. So you can do this with any of your data. This is a really useful tool if you're looking at different species, for example, right? Like, and someone said, what was the layer you mentioned for phylogeny? ggdendro. Yeah, I believe it was ggdendro. It's not a layer, it's a package. Let me check all the packages I have. There's so many, but there is one. Most of the ggplot extra packages are GG. So you can see how many extra. Oh, ggtree. Here we go. That's what it is. So ggtree is an R package for the visualization of tree and annotation data. That one requires a little bit of work to figure out. I should actually do that. I'm going to add that to I'm adding it right now to a webinar idea. Do one just on ggtree because I use it often. And yes, webinar 3. And there is a little bit of a learning curve, but it is phenomenal. It is such a useful package. But of course, we can see here I have a few other packages for making ggplot2 publication-ready plots. This one I think adds a lot of themes. There's so many. I definitely don't have all of them. But yeah, so I mean, that's kind of the beauty of R. If you want to do something, there's probably a package that makes it a little bit easier, and there's probably 10 packages that make it a little bit harder. So. Yeah, ggplot is it is a little bit of a, I mean, there's a learning curve, right? And you do need to work with it to the best of your ability. And but I'm hoping that this webinar made it a little bit easier. And I mean, I'm totally available to answer any questions. Yeah, no, I'm happy. And yeah, no, please, I will do that one. Because I'm trying to do a little bit more phylogenetic methods, so I think a ggtree specific one would be really useful. Because it's not as ggplot. You have to add in extra data on top of your data. And I think, and yeah, Isabella, to answer your question about the manipulating data sets thing, I think we should be doing another data one about how to just clean and process data. That's something I've been meaning to do for a good while and do it a little bit more in depth. So we'll definitely be doing that one. Maybe a week after next, honestly. I think that's a very useful data set, like how to develop your own data cleaning pipelines and whatnot. But yeah, no, I mean, thank y'all. Like, I enjoy putting these on, partially because of y'all. You're nice. It's fun. I like providing something. Pretty much every webinar that I've ever produced has been something that I struggled with when I was learning. And yeah, I think especially on like refreshers, you need them often. I always find that I get better with all my tools if I'm making a webinar right. Forces me to relearn some basic stuff and troubleshoot live. Cool. Well, yeah. So look out for anything. If you RSVP and you're getting the emails, you'll know exactly when we have the handouts, when the recap is up on the website, everything like that. And if you're on the emails, you'll get a notification tomorrow about the next webinar where you can RSVP. There'll also be a post on Instagram, all that same stuff. But yeah, this was fantastic. I'm going to stop recording now. Cool. Awesome. Yeah, and I have a few free R resources up on the website. And the biostatistics textbook I have is also based in R, so it teaches you how to create a linear model in R and correlations, how to understand p-values, that stuff. And those are paired with courses. So yeah, for sure. We'll see you next week. Thank y'all for tuning in, and continue to learn adventurously. Yeah, I plugged it in. I plugged it in right at the end. Cool. I'm gonna stop streaming now. Thank y'all.